Hi, I'm Paul Graywall with Sick Venture Studio, and we're here with a panel today to talk about emerging trends in oncology. And I see a lot of familiar faces, a lot of folks from oncology, literally everybody here, and a wonderful panel here. I'll let them introduce themselves, starting with Chris in a second, and when you do that, let's try to maybe say something brief about what yourself, as well as like your current developments in cancer, right? What you're doing to um, promote whatever you're promoting. Sure. My background, I'm trained. I did training in immunology, software engineering. I built companies. I've helped launch over 250 startups. We have and currently Sick Venture Studio is a fund and a studio that incubates Stanford spinouts in oncology, therapeutics, diagnostics, AI, precision oncology. Chris.
Okay, I'm Dr. Chris Apfel, made in Germany over half a century ago, which you can actually recognize from both my accent and my gray hair. I've asked way too many questions, so they kicked me out and I had to flee to the US. Being originally an anesthesiologist intensivist, have done a lot of clinical research, published over 100 papers, have developed a clinical prediction model that's now standard of care until my dad was diagnosed with lung cancer. And I realized that there are some limitations with genomic testing that can be overcome by really testing the original patient's tumor. So we founded Sage Medic. That was not out of UCSF. I actually left the clinical department there and founded this together with my wife. And we have now been able to develop an assay where we can take a patient's live tissue, create hundreds of microtumor replicates that reflects the tumor heterogeneity and the tumor microenvironment. And then we can drug them and see until they are really drunk. And then we can, after a week, we can actually see how they're responding to it. And then our latest news is that we got a patent issued that actually allows us to multiplex cytotoxicity and antiproliferation, because some drugs are actually more antiproliferative and some drugs are more cytotoxic. We can differentiate between those, which one is actually important for tumor response. The other one is to increase progression-free survival. And we can test it and have a test result for the patient within 7 to 10 days. And that's a big deal. We are CLIA certified, fully accredited by CMS. It's an LDT, a laboratory developed test, and which apparently doesn't need FDA approval. And it's now available. So if any of you is planning to get cancer, just talk to me.
Hey everybody, I'm Julianne Averill, and my background is I'm the finance person on this panel. So I work for a professional services company, Stanford Advisors, and what we help companies do is they think about if they have a strategy, they help turn it into action. And so that's action on the financial operations side, the clinical side, and then even market access. And so my companies, they tend to take their products and they can do anything from have an AI platform that focuses on target discovery and then moves into the clinic, or they're even focused on figuring out what does that precision diagnostic look like. Or we even have companies right now that are in the telemedicine and longevity space, figuring out how do we actually help the patients in times of need. And how I got started in this space was actually when I was 10, I worked with my parents in their medical clinic helping on the ops site. So I actually was helping my parents figure out how do you do the doctor's clinic side. And then where we are today, I also serve as an audit committee chair for Cure Search, where we help find cures for children's cancer fast.
Hi everyone, I'm Christabel. I'm a new faculty at Stanford in the Department of Radonc. My expertise is in cancer immunology and the tumor microenvironment, and my goal is to develop therapeutic strategies, especially immune therapies to target, innate immunity, so inflammation, fibrosis, and the crosstalk between cancer cells and the innate immune milieu. We're spinning out our company, hopefully, within the next 6 months from Stanford, that we've developed a recycling antibody to treat ovarian tumors as our first indication. So yeah, happy to be here and answer any questions, and look for opportunities to work together.
That's a hard act to follow. I'm Charles Keller. I'm a pediatrician and a children's cancer doctor, scientific director of CCTDI, a Children's Cancer Therapy Development Institute, a nonprofit biotech spinout of Oregon Health and Science University. So we do basic science. So I've been NIH funded for 23 years. We have a Cancer Research UK Cancer Grant Challenge grant actively too. And the work we do is related to antibody drug conjugates, small molecules, and protein degraders, which includes the work we're doing with Nurix Therapeutics in Mission Bay. They're really heroes of pediatric cancer, trying to develop 5 new drugs for childhood cancers in markets where there might only be 400 to 450 kids per year in the United States with those cancers.
Okay, thank you. So maybe I'll start with a question coming back to Chris Opfel over here. And so you have a platform and a new platform to test and to do testing. And then what do you think, can you talk a little bit about further developments that you see beyond what you're currently doing, what you see out in the market, and where you see the direction of going? For example, if we all started to, you started to test everybody the similar way.
So I think what is clear is that with our platform, we can test tumor tissue independent of its original primary origin and independent of the presumed mechanism of action. And so that is applicable for a wide range of solid tumors, especially in the late stage, when they also have mutated and have developed their own sensitivity and resistance profile, and you cannot know this until you have tested it. And it's also interesting especially for rare cancers, so pediatric cancers, but also the whole field of rare cancers where there are often no large clinical trials. That's actually where the Sage Oncotest is useful. The other part is, since drug development was mentioned, we can also test drugs that are planned to be taken into the phase 1. So instead of a PDX model, we are basically faster, cheaper, and better than a PDX model because we have the original patient's tissue. We culture this in the own patient serum as well, and we can then distinguish between drugs that are more likely to work on primary tissues, and that's actually the important part, to work on primary tissues. One often says, well, a mouse model doesn't translate to the human, or a petri dish is even further away. If you put a cell line into a mouse, you still have the result of a cell line. And so it's actually, the key is what's the primary tissues that you're looking at, and that's actually SageMatic's core characteristic, to use patients' fresh, original tumor tissue to make a prediction.
Okay, Chris. So a lot of people have tried this. There's like 20 companies on the market, 10%. Why are you succeeding?
Well, the previous companies, so we have actually looked into this very carefully before we started Sage. And so first of all, there is the assumption on oncologists, it has never worked. Actually, if you look at the data, and we have done our own meta-analysis, there's actually an 80% sensitivity and specificity to make a prediction. And one can actually double the patient's chance of a tumor response by understanding what is the therapy that's more likely to work, number one. Number two, there were other companies around that, for example, were growing tumors. So the clonogenic assay and organoids, in a way, are part of this group, which is heavily funded by the NIH for many decades. But organoids need time to grow. They need weeks and sometimes months to grow. And low-grade tumors often don't grow. So then you don't have a result. And so the advantage of the Sage Oncotest is we actually are not an organoid model. We take the original patient's tumor and are not dependent on the tumor to grow. That's one of the advantages. then tumor microenvironment, then we can actually measure cytotoxicity and anti-proliferation. So there are many things that only we can do.
Okay, wonderful. Okay, you want to talk about climate? You want to talk a little bit about sort of the finance side and emerging trends in finance and so forth, right?
Sure.
Okay,
Yeah. So the emergent trends in finance. So right now, for example, with companies, what we're seeing is, as we've talked about earlier, there's a lot of innovation and there's a lot going on with these new and emerging technologies. However, as we try and figure out how do we fund these companies and fund these technologies and bring them to market, it's been a bit challenging. So what we're finding is early stage companies to late stage companies are trying to figure out, okay, what do we do now? For example, we're seeing changes and struggles with the NIH, governments, and what do we figure out? And so from that perspective, I'm starting to see a lot of my organizations as well as additional organizations move towards creative strategies. And what those creative strategies are is you align your product and your fundraising strategy with basically the funder's need, as well as, like we mentioned earlier, the capital efficiency. And what I mean by funding need is you think about partnering with investors where they actually have a focus on patient outcomes, as well as some kind of alignment around either location or product. And so, for example, at Cure Search, for example, since the focus is on finding cures for children's cancers fast, if your particular product can be converted to a therapeutic for children's cancer, then you actually have the ability to partner with them on grant funding there. Or additionally, there's funding opportunities with the state of Texas. There's also international funding opportunities with Saudi Arabia, as well as international with Italy. And there's different opportunities that can be found as long as you're willing to have your organization partner with those organizations and those countries as well as those states.
Okay, so lots of different new emerging finance opportunities. Okay.
Yep.
All right, so what's emerging at Stanford?
A lot. I think I don't want to comment on every innovation from Stanford, but so I'm a huge fan of immune therapy because I think we focus so much on just targeting cancer cells, and it's not just that, right? You have the entire, there's a crosstalk between your mesothelial cells and your cancer cells and your macrophages, and developing therapies that can efficiently target this crosstalk, I think, is the future, and a lot of labs at Stanford are working on this. Also, I think chemoresistance is a big issue, and as Chris's company is working on developing tools to test what are the best therapeutic strategies. I think developing combination therapies, which are immune therapies which can overcome chemoresistance, is the future, right? And that's what we're working on, and I truly believe if the science is sound and robust, speaking like an academic.
Can you double click on that? So what does that look like when you're combining those two? What does that therapeutic approach look like?
Yeah, so for example, what we're working on is ovarian cancer has the biggest issue is cisplatin resistance for patients, right? And where our next indication is HCC and other solid tumors. But we want to prime cold tumors, to make them into hot tumors where they're easy to target. So we're not going to take away, chemotherapy, but what we want is to give our tumors immune therapy. So the therapeutic we're working on, we're trying to block the crosstalk between mesothelial cells and the cancer cells. And so when we block this, we chemosensitize our cancer cells to cisplatin therapy. So the patients will be given a much lower dose, so cytotoxicity won't be a problem, and you're priming the patient's immune system, right? And so in a way, this is, not just a short remedy, but something that can improve the quality of life of the patient. Not just they're not just surviving, but also having a good life 10 years later. So that's.
So is that combining immunotherapy and oncology? Is that immuno-oncology?
Yes, so that's immune oncology, right? Because we're trying to elicit a response from the immune cells. So we're blocking this interaction between in our case, mesothelial cells and cancer cells. But we're also developing other therapies that can prime macrophages, for example, to attack, your cancer cells. So the answer here is, can we develop methods, right, which can prime your immune system, inhibit any interactions with the cancer cells, where the immune system is, like tumor microenvironment tells your cancer cells, right, like grow. And so you want to block that interaction. So it's kind of like an orchestra, right? Like I think of solid tumors like a, very well functioning orchestra. And, you want to block this signaling. And the way to do it is using, in my opinion, my humble opinion, antibodies. And right now, I think recycling antibodies are the future. There was one that was approved. And the advantage of a recycling antibody over monoclonal antibodies is that they bind to, your antigen, and they degrade it actively. So when you have a protein that is highly expressed in your serum, you're not just having your monoclonal antibody bind to it and stay there. You're actively degrading this protein. And so, there's a high, for proteins which have high turnover rate, you're getting rid of them in like real time. So, yeah. So.
Okay, wonderful. Okay, next question. Can we double click? You mentioned a little bit about ADCs and small molecules and your approach. Can you talk about a little bit about what's emerging and why you chose those modalities and what else is out there?
Sure. Well, so childhood cancer is the beneficiary of a lot of wonderful people in pharma and biopharma who are looking for simple single gene diseases where they can get proof of concept that their drugs work. And so I think one of the really neat advances these days, and especially since we're here in Palo Alto, the Silicon Valley, I come from the Silicon Forest, just outside of Portland, Oregon, where there are R&D facilities for Intel. So computational drug design. And so, we all hear about machine learning and artificial intelligence, and AlphaFold 3 is real. It's fabulous. You can do a lot of prediction of protein structures. You can do a lot of drug docking. You can make those drug dockings not just static, but you can heat and cool those computationally and find new drugs. Lawrence Livermore National Labs with BioBridge have developed a KRAS inhibitor drug, which has come to the clinic and is very exciting, just showing the power of computation. And sometimes those computations aren't machine learning or artificial intelligence. Sometimes they're just because we have big computers and we can do actual molecular modeling, even quantum mechanics, which is the side gig of Lawrence Livermore. So computational drug design is exciting, and it's here, and we're able to drug now not just enzymes, but curiously, transcription factors. So that's something exciting to talk about. Some of the other things that are going on, well, you can take those small molecules that you might be able to dock to a protein, and you take them right to the recycling bin of the cell. So protein degraders, molecular glues. If you remember from the 1960s, for people who are about my age, a number of people, their moms took a medicine called thalidomide, and so they have quite short arms. But that we've come to figure out was a molecular glue, and so you can use the same technology, the same principles, to take a driving cancer protein and put it right in the recycling bin. And so that's what Nurix Therapeutics is doing, and they've had some really nice public data on their BTK protein degrader. And so our Venus has an estrogen receptor degrader that they did a phase 3 trial, which in a subpopulation had nice activity. So take a look at that, both publicly traded companies. I'm not saying anything other than the public information. And then I think the really neat thing, because it's kind of having a moment, are antibody drug conjugates, and those drugs might be chemotherapy. We finally figured out how to link an antibody to a drug payload with a linker that doesn't fall apart. But those payloads, they can be chemotherapies, things very traditional like topoisomerase inhibitors, or things that are very novel, like DNAs that interfere with the expression of genes in things like myotonic dystrophy, which is a disease that certainly needs a lifelong treatment. Might be rare, but if you look at the multiples of the patients taking that treatment every week for the entirety of their life, sort of the Genzyme model, there's a lot of money to be made by investors who will pick a rare disease and aim to cure it.
Okay, wonderful. Thank you. So, Christian, ovarian cancer came up earlier. And Chris, you have a use case right for ovarian cancer. Can you tell us talk a little bit about that use case?
Sure. We have dozens of use cases. Actually, we published at the AACR, presented actually our novel multiplexed cytotoxic and antiproliferative assay, where we actually were probably the first one who actually found that there are certain drugs that really inhibit the growth without really killing the tumor. And that's not necessarily the same information. So PARP inhibitors, which are currently the big cost burden in the ovarian cancer space, are actually primarily extending progression-free survival, usually sometimes by years. So it's a real game changer, but only for a certain number of patients. And you don't see this in a cytotoxicity assay, which other companies are always doing, but only in our antiproliferation assay. There you see actually big differentiations in terms of PARP inhibitors. And the other drugs that actually inhibit growth and proliferation are the taxanes. So we can actually see whether there is actually another drug, an alternative drug, that's actually a generic, inexpensive paclitaxel, docetaxel, that may actually be more effective than a PARP inhibitor that costs $100,000 or $200,000. So that's kind of one in general observation. Interestingly, we had yesterday a conversation with a patient who presented with ascites as first symptom, so fluid in the belly. We actually got the ascites. There was quite a number of cells in there. We were actually identifying this. We saw very high proliferation. So this patient should have, in my mind, gone to surgery as quickly as possible. And the oncologist elected to go for the neoadjuvant therapy. So the idea is you give some therapy first to shrink the tumor so that you have better conditions. That's been quite popular in the United States, not as much in Europe. And I said, okay, so there is some sensitivity to cisplatin, but not to paclitaxel. If after 2 or 3 cycles it's not really responding well, then actually one should actually move swiftly towards the surgery, and then the other PARP inhibitors, which had showed some effectiveness. Got a call yesterday. The patient had 5 cycles of neoadjuvant therapy. The CA-125 didn't go as down as we would have liked to. So she had really 5 cycles of therapy that wasn't promising in the first place. And this tumor, it was a tumor that has very high proliferated quite strongly. So that's a case where the oncologist had that information, but actually didn't really act on it, and wasn't really, in my mind, on top of things to say, okay, the assay has shown this patient's tumor is aggressively growing. It may have some sensitivity to cisplatin, probably not to paclitaxel. Let's try the neoadjuvant, but if the neoadjuvant doesn't work, then rather than making the patient suffering all the side effects, let's actually then make sure this patient gets to surgery and maximize the outcome.
Okay. All right. So we only have 5 minutes left, so we'll go for a couple of quick comments left. And do we have a Q&A after it? No. Okay. So.
Yeah, so I think I would just add too with the comments so far is the other thing I'm really excited about with the future of technology is this intersection of therapeutics and diagnostics. Like, for example, I have a company coming out of the UK that is blending their diagnostics engine, which is an AI platform like we were talking about earlier, and they're using the diagnostics from the AI side to identify potential tissue, take that through the drug development phase, and then use the data from the drug development side to then feed the
Are
diagnostic.
you talking about biomarker-driven drug discovery?
It is, yes. And so it's been really interesting to see all of that. And so what I'm excited about is as these technologies continue to evolve, like seeing the integration and intersection of our different areas.
Wonderful. Christabel?
Yeah, I think there's 3 things we need to focus on, right, to overcome challenging diseases. So one is early diagnostics, because people don't know if they're sick, right? Second is better therapies, and third is effective testing systems that can recapitulate the tumor microenvironment or the disease. And I'm hoping that all of us here in the room are working towards each of these, and we can combine them together to solve the big problem of, overcoming these diseases.
Thank you. Sure.
So as news you can use, and as a plug and an advocate of kids with cancer, it's kind of amazing the resources that are available to startups, but also investors when you think about the companies that you support. So in childhood cancer, if you enroll in a national clinical trial, they'll check for any common mutations. And they'll return those results not just to the doctor, but to the patient within 6 days. They've open sourced all the information that's available for childhood cancer trials through the University of Chicago program. And you can just sign up with an email account. You don't have to have an affiliation, just promise to use the data properly. Um, Foundation Medicine's outsourced all the mutational data from kids that have relapsed from childhood cancers, and Champions Oncology makes all of their model information for the patients that have had new diagnosis or relapsed patient-derived xenograft models available for free. They'll even share the mice. So it's just a wonderful place where if you need proof of concept and you need consistent data on a national level, you can go to these resources and you can prove out your platform and you can show your value proposition.
Okay, wonderful. And speaking of early detection, I'd like to say the Grail folks are here somewhere, right? So I just want to thank for being out there, and then you guys should really consider taking that test. I mean, it's probably the top test in the world right now for early detection. Gallery over here, and then also I'd like to say thank you to Roger for putting all this together, and Ari and Brad and folks. All right, thank you.